So as promised yesterday, we're going to talk about neurovariability. But also I wanted to take you on a little journey. So I do a lot of language research. So I look at the neurobiology of language, and we're going to come back to that later in terms of variability. But a brief historical vignette. This is not working. Just worked a second ago. Okay, so I'm originally from Munich, and most of you probably know Munich. What's going on here? For the Oktoberfest. But that's not all we do there. Well, we do drink a lot of beer, but we also have a beautiful anatomical institute, and that is where I started learning anatomy. So it's in the heart of the city. Pettenkofer Straße, exactly. It's a beautiful building. And that was when I started falling in love with the actual brain and how to study the brain. Now, when you look at my trajectory in Munich, this is where I grew up. I moved a little bit, then decided it's time to cross the river, go on the other side. Enjoyed my time there, but then really you always go back to where you came from, right? So I went back. Now, if we look at my trajectory within the city and the city overall, then I didn't really had any other choice than study the neurobiology of language. So it all begins with anatomy, but there is not just one type of anatomy. There's different types of anatomy, and in the neuroimaging world, these are examples where we can look at the surface, the gyri, the sulci. We can cut through the brain, look at sectional anatomy and the differentiation between the gray and the white boundary. We can look at the white matter connections, the connection anatomy, and we can put people in a scanner and get colorful blobs on the brain to look at the functional anatomy. Now, what's important here is that different parts of the brain can be referred to with, the same part in the brain can be referred to with different names depending on what kind of anatomical labels you're used to. And then personally, I work a lot with neurosurgeons. They don't per se have a different anatomy, but they have two things that are different. The first thing is they turn everything upside down. And the second thing is that they orient themselves by vessels more than gyri and sulci. So there's not just one anatomy, and when we talk about anatomy to each other, it's always good to make sure that we have a common ground. Now, speaking of common ground, this is a question I love asking any audience. So I'm going to ask you as well, very briefly, how many lobes does the brain have? You get a second to think about. I give you numbers, raise your hand. 1, 2, 3. We're not hackling here. 4, 5, 6, 7, 8. I'm running out of numbers now, but hands are still going up. So the pattern that you see here is a pattern that we always see, and that is that we do not even agree on how many lobes the brain has. And part of that is because it changed over time. So those of you who said 4, you're still in the 19th century. Those of you who said 5, welcome to the 70s. And those of you who said 6 are in the 90s. Now, the definition has changed, and there's still a lot of debate, but this is just to show you that even on the most fundamental principles of anatomy, we don't necessarily agree all the time. Now, we've been talking a lot about big data and AI, so I thought I'd ask AI and Google if they know the answer. So first time I asked was in May 23, and Google back then said it's 4 lobes, 19th century knowledge. But I'm persistent, so I asked again in September. Someone published a paper, and Google updated and now thinks it's 6 lobes. But then Google is also good, right? So I asked ChatGPT. ChatGPT is interesting because it depends on how often you ask it. So the first time I asked, is that 5? Then I literally just repeated the very same prompt, 4, repeated the prompt again, and it said 6. So the lesson here is ChatGPT will give you the answer that you want or need if you just ask it often enough. So AI is not ready to teach anatomy, is the conclusion. Okay, now the next question is, how many white matter bundles do we have? So here's a beautiful illustration from Alex Lehman's. And you can see the whole brain tractogram and the connectome of the brain. And you see that just by looking at it, it's quite difficult to differentiate individual bundles. And that is because the separation is not necessarily clear. And we heard yesterday that if you know the trajectory and the end points of the tract, you can dissect them out. But if you don't know the anatomy, it's a lot harder to dissect out the individual tracts. Now, for the human brain, there's only really two methods you can use to look at the white matter and dissect the tracts. One is highly invasive postmortem Klingler dissection, and one is diffusion-weighted tractography. And you heard a lot about the method of tractography, so I don't have to go into too much detail. So I decided to give you a little rundown about Klingler postmortem dissection. And there was a beautiful paper published in Brain Structure and Function about the history and the method of how to preserve the brain to do postmortem dissections. And the only thing that I want to say at this point is it does take a lot of time. This is not something you can rush. The brain takes about 3 months to be prepared, and you have to constantly freeze it and thaw it. Iteratively, it does take time, but it's worth it, because if you prepare it the right way, you have a beautiful specimen to work with. Now, how does it work? Just to let you know, this is not how we do it, but this is the only video that I'm officially allowed to show. So you start by carving away the cortex, you expose the white matter underneath, and then you use tools, in this case a spatula, to remove the cortex, expose the white matter, follow the white matter pathways, and you start seeing them appear here. It looks quite brutal in this example. You see the putamen appear, and now you beautifully peel away the cortex to expose the radiating fibers underneath the cortical layers, and now we see the different white matter connections. Now, interestingly enough, and I learned this over the last month, depending on where you are in the country, the tools we use are different. So in Europe, we use a wooden spatula, but I learned that in Korea, they tend to use metal spatula to do the dissection. Quirky fact I didn't know about. This is what it looks like when we do it. So you need a good specimen, you need a bit of time. These are the instruments that we use, so nothing fancy. The spatula we carve to the shape that we want them to be in. And then optional, but recommended, is the use of a microscope. Now, when you do that, you start from the surface of the brain. You identify your landmarks first, so just to give you a few pointers. And then you start by dissecting out a C-shaped structure that goes around here through the parietal and then along the frontal lobe. That's the outer C. You start carving away the cortex and you expose the white matter underneath. Now, at this point, depending on how good your anatomy is, it gets a bit harder. So I'm putting in some landmarks again. But you see, one of the problems of the technique is that we cannot really see anymore where the white matter is going to, because by virtue of the method, you have to peel away the cortex. A few more landmarks here, and then we can go deeper. So we tend to do a progressive dissection from the lateral to the medial side, and then from the medial to the lateral side. So different to the video that I've shown you, where they just take out the insula, basically. So now we have the arcuate fasciculus here, arching around the sylvian fissure, and then we go one level deeper. So now we cut the arcuate fasciculus here, and we expose the white matter underneath, which is the inferior frontooccipital fasciculus, the radiations of the internal capsule, the uncinate fasciculus, and so on and so forth. And you do that progressively until you exposed all the white matter of interest. Now, when you do that, and this is from just 2 weeks ago in Zurich, where we do the white matter course every year, we see at least 15 brains every year. And what you notice very easily is that every single brain is different. Every single brain. No brain is like the other. They all have similar features, but they're not identical. And this is a beautiful atlas by Ono, where you can also see that some of the sulci structures have a very reliable incidence rate, whereas others are a lot more variable between people. So this is something that we know from postmortem anatomy. We know the brain is different. Now, here comes tractography. So the first thing to know is that not every diffusion-weighted imaging acquisition or every type of tractography is DTI. There's differences, and you had beautiful introductions to the method. Here is my very quick recap. So we have a tensor shape that is indication of where the diffusivity of water molecule goes. Based on the shape of the tensor, we can produce maps, mean diffusivity, principal eigenvector, FA, color-coded FA. And if we zoom into each voxel, we have a principal orientation per voxel, and then you basically piece it together and you do a fancy reconstruction. That's the fundamentals of tensor imaging. Now, the reason it looks the way it looks is because of Wendell Creek. He published a couple of beautiful illustrations where the white matter is shown in these tubes. And we had a copy of that in the library in London as well. And that is the thought giver of the visualization of tractography. Now, what's important to know is that it's a visualization. We're not looking at individual axons because the resolution is way too coarse. Now, tractography, as we have heard over the last days, has limitations, and you see one of the limitations right here. So we have the medial reconstruction of the corpus callosum, we have the corticospinal tract that comes here, and we know there should be lateral projections, but we cannot see them with a classical tensor model because we cannot resolve the crossing. However, there is advanced models that actually let you resolve the crossing and let you track through the corticospinal tract. Now, depending on which kind of method you use and what kind of fiber populations we're looking at, with the classical tensor model, if you have, 50, 70, 90-degree crossing, you just get one orientation. But with the advanced models, you can resolve the crossing fibers to a lot smaller angles. Now, that is good, but it comes with some caveats. So if we do different types of tract reconstructions, what we see here is the variability between the algorithms, and this is a neurosurgical example where you have a tumor here, the corticospinal tract here, and there is a couple of groups that use the distance between the tumor and the white matter as a safe zone for the reconstruction of a tumor. And you can see that if you change the algorithm, that safe zone is a lot smaller. So the choice of the algorithm makes a huge difference in the clinical field. Likewise, coming back to language in the brain, the reconstruction of the language network changes because the arcuate fasciculus in all the segments in classical DTI barely reaches the anterior inferior frontal gyrus, whereas with spherical deconvolution advanced models, it overshoots the classical language areas. So it's important to know what kind of tractography you're working with. So, coming to language in the brain. This is the classical model and the one that I've shown you on the Munich map, where we have three types of areas in the brain. We have this area in the frontal lobe, Broca's territory, or the inferior frontal gyrus. We have the posterior superior temporal gyrus, or Wernicke's area, and then we have Geschwin's territory in the inferior parietal lobe. They're all interconnected by the arcuate fasciculus, either directly through the long segment of the arcuate fasciculus, that's the frontotemporal connection arching around the Sylvian fissure, or through an indirect route, there's the anterior segment and the posterior segment that relay in the inferior parietal lobe. Now, I'm going to spend my last 20 minutes to show you why this model is incorrect. And I want to start with this beautiful example that we've seen yesterday, quite an iconic map. And here you see the visual system and the auditory system. Every single box is a region of the brain. Every line is a connection between these regions. Now, if we all agree, and I think we do based on yesterday's talks, that the visual system and the auditory system are as complex as these diagrams suggest, and surely something as complex as language needs to be a bit more complex in the brain than this model that we see here. Now, tractography and other clinical measures like direct cortical stimulation, for example, have helped a great deal to enrich our understanding of language in the brain, moving from the classical 19th century arcuate model to including the indirect route through the inferior parietal lobe to an extended language network that connects many more areas in the brain that are relevant for language function. Another thing that is a problem with this model, and it's this one. So any idea who these ladies are? You two don't get to say a word. Anyone else? Take all the time you need. What do they have in common? I don't need you to specify the names, but yes, Susan. They all won the Nobel Prize. In fact, these are all the women who won the Nobel Prize for medicine and physiology. Now, we're going to come back to that. So, I actually have all their names, but in the interest of time, I asked my student to generate an image of what you need to look like as a woman to win the Nobel Prize. And this is the average Nobel face. Now, there's a couple of things here I need you to take away. The first one is that the image in the middle is a face, so the algorithm worked, right? We have 2 eyes, a nose, a mouth, great. But it doesn't look like any of the individual women around it. Nor do any of the individual women around it look like the average face. Now, here's a trick. If you want to make it into the average image, wear leopard. Where is she? Here. French leopard print gets you into the average. But the point that I want to make here, and it's plain obvious when you look at faces, is that the average image doesn't really represent the individual. And it's very evident when we look at faces of people, but it's a lot less evident when we look at the brain. So this is the average brain that we're working with, MNI template. And we can see areas where we have a good agreement between people, it's quite sharp boundaries, but areas where we have less agreement between us, the boundaries become blurry. And here's 2 concrete examples. So the motor cortex, classic textbook teaching will tell you you identify the hand knob area by finding the inverted omega sign. But in fact, when you look at individual brains, you can have an omega, but you can also have an epsilon or even a straight line. It's not there in every brain. And when we look at the white matter, these are 2 healthy students randomly drawn from our sample in the Donders. Their white matter looks very different. And if we look at the uncinate, for example, it's extremely prominent in this student. It's a lot less prominent in this student. So there's a huge degree of variability in the cortex and the white matter. Now, we wanted to look at this more systematic rather than show you hand-picked examples. And this is work that I did with Michelle, where we plotted the variability across the population for the gray and the white matter. And what you can see here is that there's a gradient of variability. So the deeper parts of the brain, the older parts of the brain, tend to be more alike between us. And as we move out, there's an increase of variability. Now, that's the group average, obviously. And we also looked at the impact this type of variability has in a clinical setting. And here's an example from surgery again, where we identified the tumor in the brain and dissected the individual corticospinal tract right here. But then the question is like, if we were to use an atlas, where would the corticospinal tract be? And the atlas said it's right here. So this goes to show that relying on Atlas in a clinical setting can have detrimental consequences for the individual patient. Now, the next thing we were interested in is, okay, so there is variability between us, but there's also variability within each brain. So Lilith Dulyan, a PhD student in my lab, looked at the variability across all the tracts of the language network within the left and the right hemisphere. She looked at the asymmetry between the hemisphere, and we can see there's a pattern whereby some tracts tend to be more left lateralized, others slightly more right lateralized. But she was also interested overall which hemisphere is more variable. And it turns out the left hemisphere for the language connections is more variable than the right hemisphere. And if we bring it back to the individual tracts, what we can see is that the anterior segment of the arcuate fasciculus, the uncinate fasciculus, the hook between the orbitofrontal and anterior temporal, and the IFR fasciculus tend to be most variable in the left hemisphere. Okay, so now we know we're different. Now we know there's a tract-specific pattern that varies across the hemispheres. The question now was, does it matter? Does it matter that we are different? And this is a study I did with Patrick Friedrich and Henrietta House and Michelle, and it was supposed to be a quick study. Turned out it took a year to review all these papers because there was 326 studies that looked at the variability in the white matter. And here's a couple of things that we learned. The first is that most of what we know about the function of the white matter, we actually know from clinical populations. So about 45% are from neurology and neurosurgery, 29 psychiatry, and only 25% of the studies were conducted in healthy participants. The other things that we learned is that some tracts are extremely prominent, like the corticospinal tract, whereas other tracts are a lot less prominent, and that varies across the domains. Now, we looked at the association between the variability in the anatomy and the variability in cognition and behavior. And here is the interesting thing that we saw, and I'm going to zoom in to the arcuate as an example, but this pattern holds true for all the tracts that we looked at, and that is that there is no one tract, one function association. So all the tracts were associated with several cognitive or behavioral measurements. Okay, so now we know we're different. Now we know it matters for healthy cognition and behavior. What do we do with it next? So the question is, does it matter in a clinical setting? So we recruited acute stroke patients who had a lesion to the left hemisphere. They lost the ability to articulate or communicate. And I'm going to come back to that. And we plotted the arcuate fasciculus in the healthy right hemisphere and their language recovery over time. Now, when we do that, we have the arcuate on the x-axis, the language recovery over time, and I'll highlight 3 patients for you. Patient number 1 is down here. So his recovery 6 months after symptom onset was not great. And when we look at the arcuate fasciculus, it's a thin connection in the right hemisphere. Patient number 2, that's slightly better, and we can see the connection is slightly thicker. Patient number 3 has a really thick, strong arcuate in the right hemisphere, and she, according to the Western Aphasia Battery, completely recovered back to normal language functions. Now, what is interesting about the study is that if you just look at clinical and demographic data, then the predictive power is about 30 to 40%. By simply adding the variability of the arcuate fasciculus in the healthy hemisphere, we can boost that prediction to nearly 60%. So that's a huge gain in explaining the variance in recovery based on the variability. Now, a classic example for a disconnection aphasia is conduction aphasia. Ever heard of conduction aphasia? Some in the room. So the idea with conduction aphasia is that you have the inferior frontal gyrus that is important for articulation. You have the posterior temporal gyrus that's important for comprehension, and they are connected by the arcuate fasciculus. So if you have in the classical model a disconnection of the arcuate fasciculus, then you're able to articulate because the inferior frontal gyrus is intact. You're able to understand because the temporal lobe is intact. But what you cannot do is repeat because you cannot send what you heard along the arcuate to articulate. Classical disconnection syndrome. Now, this was originally proposed in the 19th century, and that has been the model ever since. But with the emergence of CT and later MRI imaging, there was more and more cases of patients who had a lesion to the inferior parietal lobe, not the arcuate fasciculus, and they presented with the same clinical symptoms. So now that story shifted towards the inferior parietal lobe rather than the arcuate fasciculus. Now, this was a conundrum that we couldn't solve in our stroke patients, so we changed and studied primary progressive aphasic patients, where the aphasia is slowly oncoming rather than abrupt disconnection of the white matter. And we had tractography data and cortical morphometry measures to track the atrophy in the cortex and the white matter. Now, when we looked at those patients, what we could see is that the arcuate fasciculus, the long segment, the classical arcuate, had absolutely no correlation with repetition deficits. The indirect segment, on the other hand, through the inferior parietal lobe, did correlate with repetition deficits. When we looked at all the areas associated with language functions, the only area that survived multiple comparison correction was the posterior supramarginal gyrus right here. So again, pointing towards the inferior parietal lobe. So what this data suggested is that after 150 years of considering conduction aphasia as a disconnection syndrome of the arcuate fasciculus, we took the arcuate out and proposed a new model whereby a lesion can occur along the network, disconnecting the anterior or posterior segment or damaging the inferior parietal lobe, and it will result in repetition deficits. Now, depending on where in the network the lesion occurs, you will have a different flavor of repetition deficits, but it's not a classical disconnection of the arcuate or just an isolated cortical lesion to the inferior parietal lobe. It's a network disorder, and a lesion can occur anywhere in the network to cause symptoms. Now, I was extremely happy to see in a very different context that George Samadouris in the UK operated on a patient awake, and they did some language assessment in the patient. And when they stimulate the parietal lobe, the inferior parietal lobe, they actually induced repetition deficits. And I hope this plays.
She's fluently having a problem there. Yeah. No mix and full buttons. Okay. So actually we get one you put that on 4, yes? Or the 5 repetition. That's DCS, yeah? Yes. House. Okay, let's try again. Today is a nice day.
So you get the idea. Every time the inferior parietal lobe was stimulated, that patient was unable to repeat what he heard. And that really nicely matched with the data that we've seen in primary progressive patients. Now, Nicola yesterday said something along, I wasn't quick enough to write it down, but something along the lines of no method is perfect and we should combine them. And that's what we did. So here we used classical functional MRI data. We used the disconnectome data that Michelle presented earlier today. Got the disconnectome component maps. We put it together and statistically ran a regression over it to get the beta values, and that would let us identify the white matter map of significant contributions to the group level maps of reading in fMRI. Now, the results, when you bring it all together, is that we have our activation map for the fMRI activation. we were able to identify the critical white matter connections relevant for reading deficits. So here we have the vertical occipital fasciculus in the occipital lobe, and then here we have U-shaped white matter fibers around Exner's area in the middle frontal gyrus connecting the precentral gyrus. And then down here we have a lesion that would cause a reading deficit. Now, what you see here is that by combining the different modalities that we can get from imaging with clinical data, we can actually dissociate the maps that we need to better understand clinical presentations in patients. Now, where does that leave us? So this is the classical model. I've shown you various examples of what I believe this model is not sufficient to explain the complexity of language in the brain. And Michelle and I recently wrote a review about this, where we proposed a new model. So you have the classical model from the 19th century, in plus Geschwind's territory that wasn't there in the 19th century, where you have a modular understanding of language in the brain. So one area does one function. Inferior frontal lobe does articulation, posterior temporal lobe does comprehension, so on. Now, this model is insufficient to really capture the complexity and the dynamic of language when we're talking, especially in interaction, talking to each other. Now, the other model is the hierarchical model, where these areas are interconnected, for example, by the arcuate fasciculus, and repetition is a very good example of that, where the information has to arrive in one area first and then gets propagated to the next area. But again, this model is insufficient and doesn't capture complexity of language in the brain. So what we proposed is more that language is an emergent function of the brain, and it emerges through the interaction, the dynamic activation of different networks and states in the brain that will let language emerge. And it took me a while to really capture the concept properly and come up with a good way of explaining it. And the best example that I have for you is that if you think about language in the brain, it's akin to thinking about the music in an orchestra. The music is not in the individual instrument, it's not in the notes, it's not in the conductor, it's in all of them coming together in a dynamic fashion that music emerges. And this is how I believe language works in the brain. And with that, I thank you for your attention.